Okay, so this is a picture of the city where I work. I do not live in Liverpool. I live some way away from there, but it's a very large city, very vibrant city. There are only two things that you need to know about Liverpool. One is the football team, and the other, of course, is the Beatles. The Beatles have not been together since 1970, but still everyone talks about the Beatles all the time in Liverpool. I don't know why. Just a little diversion before I begin. These are kind of to the same scale. So your state here in São Paulo is around about the same size as my whole country. So when we make a trip in the UK, so from Liverpool to London, that is considered quite a journey. This is perhaps the same as São Paulo to San Carlos. Very big difference in scale. Your country is enormous. But I love to come here. This is my 5th visit. I came here first in 2009 to Ribeirão Preto. I was in Ribeirão Preto again last week. I have quite a few collaborators here now. And it's a great place, Brazil, because you get photodynamic. I like that. Other places in the world, they don't understand, they don't want to know, including the UK. The UK is very difficult. So, today I will try and set the scene. Okay, so I'll talk about photosensitization, which I guess you probably will understand. Something about microbial physiology. In other words, how can we target microbes? You know, why do we target microbes and not affect human cells or animal cells? Because this is important. And also I will talk about toxicity. How does toxicity arise? How do we make cells die? How do we make microbial cells die? Or how do we inactivate them? What targets do they have? What is the chemistry? We understand a lot about the chemistry on a biomolecular level now, of course. I would also say, if you perhaps overindulged yesterday, not looking at anyone in particular, there are some clinical slides of disease which may not help with your stomach or your head. So the problem is I can't remember where they are, so I press this, it will just appear. So I apologize in advance. Through the presentations on the 3 days, I will ask questions. You don't need to worry too much about them. It's just to get you to think, perhaps not just about the photodynamic side. If we're talking about infectious disease, we need to think about infection. So, when and where was the first incidence of HIV infection? And how was it caused? Does anybody actually have an idea about that? I'm always surprised about how few people know this. Okay, we don't have a great deal of time with this presentation, so I won't kind of persist with this if you don't know. So, the answer is 1908. Normally when people give answers, they say, oh, 1980, 1975. But this has been traced back using kind of phylogenetics. And so it's, it's known that it was in Cameroon, which is in the kind of middle part of Africa on the west side. And it came from chimpanzee meat, because we know that HIV came from simian immunodeficiency virus, particularly in chimpanzees. HIV-1 and HIV-2 can both be traced to simian immunodeficiency virus. Okay, and the link between the two would be butchering chimpanzee meat. And the man involved would have had perhaps a cut on his hand or his arm or something, or a cut in his mouth, and so somehow the blood barrier was crossed. But 1908, not more recent. Why should we use photosensitizers in infectious disease anyway? Okay, I often ask myself this, particularly when I've been trying to talk to a pharmaceutical company and they think, what is this guy talking about? He doesn't know anything about drugs. There is a problem, definitely, with the pharmaceutical industry, certainly in Europe and the States, trying to get photosensitizers recognized as drugs, because people just assume that because they're colored compounds, they're just dyes, and therefore old technology and impure and all the rest of it. But no. Infectious disease caused by, as I presume you know, bacteria and viruses and fungi and also protozoa. Because photosensitizers work by producing reactive oxygen species, we can cause immense amounts of damage in these very simple cells, and therefore we can inactivate or destroy them. And this reaction is merely a chemical reaction. It's not an enzymatic thing or anything like that. And so the rate of kill is very, very rapid. So it's more rapid than even something like a biocide that you might use, and certainly more rapid than traditional antimicrobial agents. So the idea, hopefully, is that there are some cases where we have localized disease that we can apply photosensitizer and light at the same time quite easily, and therefore we wouldn't need to use conventional antimicrobials in that case. That's the idea. And the other thing to remember is that these are photo-antimicrobials, particularly if they are cationic, if they're positively charged, which means that they act against all of these target organisms, unlike conventional agents, which, if it's penicillin, for example, that will work against bacteria, perhaps, but it won't work against viruses or fungi, okay? That kind of idea. So this is hopefully useful, particularly in the case of conventional antimicrobial resistance. It's recognized around the world that this is a huge and developing problem. A lot more deaths are expected because we don't have drugs which will work. The pharmaceutical industry has kind of taken a step back in many cases from developing new agents, and so there's definitely a need to conserve the agents that we do have, so if we can replace them with this approach, so much the better. So I've just given 4 kind of headings there. So local infection, as I mentioned. Patient disinfection is a good one. But often patients will, quite healthy, turn up to hospital for elective surgery, for perhaps knee surgery or hip surgery or that kind of thing. But they may be colonized with a resistant bacterium. So MRSA, methicillin resistant Staph aureus, is one of those things. It tends to kind of colonize the nose. Those sorts of patients quite often will get a post-op infection that they've caused themselves because the MRSA, by hand, I guess, somehow gets into the wound and you get post-op infection. So we can decolonize patients before they have an operation. And again, doesn't use antimicrobial agents, just uses a photosensitizer. And so they can break the kind of disease chain at that stage. We can also look at environmental disinfection, whether that's trying to clean the physical surfaces in wards, or the kind of clothing that medical personnel wear, or we can use plastics, which are used in medicine, so tubing or that kind of stuff. And we can also look, and I always talk about this in Brazil because I don't think you do it here, we can look at donated blood and the products derived from donated blood. You can disinfect those using a photodynamic approach. We do that in Europe, and some parts of North America and in China as well, so that it does actually work on blood plasma. So I'll talk about all of these things as we go on. I'm not going to spend a lot of time on this kind of thing today. I'm going to try to steer towards the disease side rather than the photophysics. But you've all seen Jablonski diagrams about the promotion of electrons and relaxation. And really, that's quite a confusing diagram, so I don't usually use that. I would usually use something more like that. So we have a promotion of an electron, and then kind of intersystem crossing, triplet state, unpaired electrons, and we can get reactions, chemical reactions from that stage. If you try and think of these photosensitizers and how they work, production of reactive oxygen species, think of chemical reaction rather than drug action, because that helps to explain why these things are so rapid. and why you don't need to target specific enzymes. We all know about electromagnetic spectrum, and the kind of tiny part of the electromagnetic spectrum that we use, and we tend to try and stay this side, the low energy side, because low energy light has no toxicity in humans or animals. We can use UV, but UV is not really a safe option if you've got tissue around. It's not particularly healthy for humans or animals. Okay, so that's where we are with the kind of range of drugs. We tend to use, or tend to work on drugs which absorb in the red, purely to avoid endogenous absorption. I'll talk about that in a minute. And there is a little bit more work these days towards near-infrared, because that increases the kind of depth of penetration that we can get. Okay, so the idea of a therapeutic window comes around from, we look at the endogenous absorption of things like heme or hemoglobin, or oxyhemoglobin in here. Melanin kind of has a fall off here. As you would expect, melanin really is produced by, we've evolved the production of melanin to avoid damage by UV, so there's not really any reason for any absorption down here because there's no oxidation caused by radiation at that stage. Once we get past that, the main problem then becomes water absorption in the near-infrared. But even so, you could get almost up to about 900 before you've got any particular interference from a biomolecule. So there's plenty of scope there for making new molecules. There is quite a bit of work, of course, on blue light absorbers. Again, I'm always a bit worried about this kind of absorption here with blue light absorbers, but they seem to work, typically in things like surface disinfection rather than deeper wounds. Just to give you a few examples, I am a medicinal chemist, so I came to this from a medicinal chemistry background all those hundreds of years ago. And you can see just some chemical examples, chemical structures of molecules here, which are all photosensitizers, but which absorb at different wavelengths, or the principal absorption, the useful absorption for treatment of bacteria or viruses or whatever. So we have curcumin up here, which is a natural product. Tends to be a fairly short wavelength blue light. Riboflavin, again, a natural product. It's around about 470 or thereabouts, I think, for that one. Protoporphyrin 9, which, of course, I'm sure everyone knows about. Methylene Blue, which is the molecule I used right from the early 90s as a lead molecule, a lead structure, and that produced molecules which look like that. Methylene Blue, I do normally say this, just in case people think I have a fixation with methylene blue, is a very safe molecule. That's why it's used in various clinical kind of procedures. It's not a great photosensitizer. So making a better molecule in terms of photosensitization than methylene blue is very, very easy. This is a phthalocyanine. This is the phthalocyanine produced and marketed by a company in Italy called Maltini Pharmacici. And this is a very useful anti-infective. You see it's a slightly longer wavelength than methylene blue. Both of those absorb in the red region. Just a note on how long this has been going on. Again, you may have been surprised by how early the HIV appeared, in 1908, but photoantimicrobials, the original Oscar Raab discovery was around about 1900, so that was even before the HIV thing. And methylene blue, in fact, was first published as an antibacterial, photoantibacterial, in, I think, 1908. It was a German paper, which I discovered not very long ago. And then methylene blue kind of occurs in the literature sporadically through the last century. And other ones will appear. The problem is that it was definitely a sporadic idea. There wasn't a great deal of interest. A group would find out something about methylene blue or erythrocin or rose bengal, and they'd publish it, and it wouldn't be picked up because, of course, by around about 1935, we had sulfonamides. 1940s, we had the penicillins, and et cetera, et cetera. So we have the, if you like, what's called the antibiotic era, which is around about from here, about 1935, probably up to about the early 2000s. After that stage, the resistance problems started to come in, and the supply of new antimicrobial agents started to dry up. So it's a kind of a double whammy there. It's not great. So we've got all this activity showing that we can kill bacteria and viruses and all the rest of it, which is pretty easy, really. It's not a difficult thing to do, compared to the damage that you need to do to a human cell, for example, to kill a human cell. It's very easy to kill Staph aureus, for example. Okay, so lots of activity at this end. This is the kind of renaissance of photoantimicrobials coming back from 1900. But the main message is that there's a lot more activity here, really, than there is in terms of conventional disease. So the argument is that we can use these, in some cases, to replace the conventionals. I'll just have a drink. So, if we look at the target size, this is a fairly rough and ready kind of schematic, but you have a human cell with all its kind of different complexities and defense mechanisms and all sorts of things there. And these are relative sizes here. And again, these are hugely approximate because different bacteria will have different sizes. There's not really an average size for a bacterium. Approximately. Fungal cells are much bigger than bacterial cells. Bacterial cells are much bigger than viruses, which of course you would know because we have things like bacteriophages, which are viruses which parasitize bacteria. So you'd expect viruses to be smaller. This kind of splodge that you can see there is a larger virus. It's a flu virus. Things like polioviruses are about 100 times smaller than that. So if I put that on there, you wouldn't see anything at all. So in terms of size, that's important. In terms of complexity, that's even more important. What we want to do is to be able to kill or destroy these things here without damaging these things here. And I should also note, because I don't think I did before, we need to think about, in terms of antimicrobial resistance, we always need to think about animal medicine, veterinary medicine, as well as human medicine. The two are very closely linked. A lot of resistance that we find in hospitals, eventually, if you trace it back, comes from practices in agriculture. Okay, so we have to consider animals, effects on animals, as well as effects on humans. And it's okay because the cell model is more or less the same. Okay? Some ideas about which targets we might think about. This is HIV-1. A cartoon of HIV-1. Important thing, really, as far as we're concerned with HIV-1, is this kind of blue layer around the outside. This is a lipid envelope. There are two main sorts of virus. One has a lipid envelope, the other hasn't. It's the one without is the smaller type, so things like polio. But most of the disease-causing viruses in humans and in animals are enveloped. And it doesn't take a very specific approach to kind of blow holes in this, or in fact just completely tear it apart. And once that's gone, then the viral particle is no longer able to infect because it can't fuse with the cell. So if we can destroy this membrane, then we're fine. That's the end of the virus as far as we're concerned. And importantly, as I say, most of the infective viruses in humans and animals are enveloped. So it doesn't matter if it's HIV-1 or a flu virus or Ebola or Zika, all of these are enveloped viruses. So we can use the same approach and inactivate all of these. This is important when I talk about blood disinfection on Wednesday, I think in the third lecture. Okay? It's the same approach. It's just a chemical reaction between singlet oxygen and the membrane. and that's it. It doesn't matter if it's HIV or flu or whatever. It will destroy or inactivate the virus. This is, again, another cartoon kind of thing of a generalized bacterial cell. Various different targets, if you like, in terms of conventional antibacterials. Okay, so if you think about penicillins or cephalosporins, they will attack specific, very specific molecular sites in the cell wall. Okay, and there's also a time limit on when that attack can occur, but I'll talk about that in a little while. So, specific drugs for the cell wall, specific drugs for DNA in bacteria, the quinolones or fluoroquinolones, things like Cipro. All sorts of different drugs will attack the ribosome, things like tetracyclines or aminoglycosides, all of these kind of things. Again, all of these are very, very specific. What is inhibited is very specific. You can't get, usually, more than one site of attack for a conventional antimicrobial. So the big difference with photoantimicrobials is we get various different sites of attack, very little specificity, and therefore it's very difficult for the bacterium to overcome that. And it's more or less, I haven't seen any studies where they've managed to find or to induce resistance through different generations. So it's obviously a multiple site of attack and highly oxidative, very rapid damage to the cell, and you don't end up with resistance. Fungal cell is a bit more complex than a bacterial cell, but still it has this kind of membrane around the outside. Most of the antifungal agents that you see in pharmacies or whatever, things, creams for athlete's foot or any of these things, all of these attack various aspects of the membrane. There are other more serious drugs which attack the kind of nucleic acid in fungal cells, but again, if you use a photoantimicrobial, you get multiple attack all over the place. So resistance to photoantimicrobials is very difficult. So mammalian cells, much bigger, much more complex, much better defense mechanisms, and so it's very easy to formulate kind of treatments, photodynamic treatments, which will rapidly kill the microbial target without damaging the human cell. So that is quite an easy thing to do. Okay, so again, a warning about clinical slides. There's 2 or 3 clinical slides coming up somewhere in the next few slides. So it's not quite as Russian roulette as I thought it was. How do we get infections? If we think about bacterial infection, then we just need bacteria kind of colonizing the wrong place. Okay, so if we perhaps if you do gardening, if you're working in the garden and you kind of cut yourself while you're digging or whatever, you can sometimes get soil into the cut. Therefore, the bacteria in the soil therefore get into the cut, and sometimes you get an infection from that. Equally, if you cut yourself normally, if there's no soil involved, there are bacteria on the skin, which are supposed to be there, perfectly healthy. Staph epidermidis, for example. If you cut the skin, you will push some skin cells through into the underlying layers, and you'll push the bacteria in there as well, so they can eventually sometimes get into the bloodstream. Most of the clinical septicemia that you see with gram-positive bacteria is caused by Staph epidermidis, which is on the skin. Okay, so if the bacteria is in the wrong place, we can cause disease. Okay, another question while you have a breather and I have a drink. Any ideas? Again, I'm not going to labor the point. So TB are around about 1 million deaths annually, but that is there's definitely a 2-part story to that. So You have the, if you like, traditional Mycobacterium tuberculosis that's very famous for killing a lot of people over a long period of time. But more recently, since the AIDS epidemic, we have a lot of death due to Mycobacterium avium, which, as long as your immune system is fine, you can breathe that in, it'll go into your lung, your body will, defense system will deal with it. If your defense system is not there, then you have a problem. So I'm not sure what the breakdown is, probably about perhaps 4 to 1 there, because when people die from AIDS, very often it's due to M. avium complex TB. Now, there may be a photograph next. Okay, we're still safe. Other infections that occur, you have obviously bacteria all the way through your system in the alimentary canal, which is supposed to be there. If it wasn't there, you can't break down your food. You can sometimes read articles which refer to the body's internal flora as an organ, a separate organ, because they deal with this with food. They break it down, metabolize it, etc. If you have oral antibiotics, then as well as trying to target whatever infection it is, if it's tonsillitis, for example, you will kill an awful lot of bacteria because you swallow the antibiotic. And if you don't kill all of the bacteria, then the ones which haven't been killed will simply grow into the space left by the ones which have been killed. So you end up with a large amount of bacteria that you don't normally have in the wrong place. So quite often that will lead to kind of stomach upsets and diarrhea and things like that. So when children, for example, are given penicillins for fairly simple ENT infections, then often they end up with fairly bad diarrhea, which is not, hopefully, not too much of a problem, and once the drug is out of the system, then things get back to normal. It can be more serious. When someone has MRSA, you understand MRSA, right? Okay. Someone has MRSA, the treatment, if it's a serious infection, if you're in hospital, you have a treatment of multiple different antibiotics to try and kill MRSA. MRSA has so many different mechanisms against conventional drugs that different cocktails of drugs have to be used. So there's this kind of blitzkrieg that occurs in the alimentary canal. What, as I said before, what antibiotics don't do, or one of the things they don't do, is kill fungi. Now, quite a lot of people have Candida albicans in the colon. And again, it doesn't do any harm there at all. If you kill all of the other bacteria, Candida will just grow. Okay, so an awful lot of people who die from MRSA, allegedly, don't die from the bacterial disease, they die from candidiasis, because there's absolutely uncontrolled growth of Candida everywhere, because there's nothing to stop it. Okay, so you get the idea. And where are we? Yes, so there is a photograph coming up next. I know where we are now. Strep pyogenes is fairly standard. It's been around for forever as far as we're concerned. It's a gram-positive bacterium. It causes fairly trivial disease, tonsillitis. Tonsillitis can lead on to other things, of course. It causes pneumonia, but it also causes, some strains of strep pyogenes cause damage to the layers of the skin. So the underlying layers, particularly the fascia. So this is where it gets its reputation as what's called a flesh-eating bug. Okay, when you read about this in newspapers, etc. So there are a couple of slides, I think. So here's one. You can see that this is on the kind of upper thigh, and you can see the depth of necrosis. This has been kind of operated on, been excised. So all of the kind of tissue on top would have been dead. And the other problem, obviously there's a big problem there because that will now need plastic surgery, all the rest, but it looks more or less as if it's okay. It's a little bit red around here, so they may have to watch out that it isn't spreading that way. But quite often with necrotizing fasciitis, so necrotizing means killing, causing death kind of thing. Fasciitis meaning the fascia layer underneath the dermis here. Okay. The other problem is that this is a very rapidly progressing disease. Okay, so it kind of eats its way through, and all of the skin is dead, and lots of toxins given out, and so it's a very dangerous situation. Quite often with this situation, you can lose the leg, because quite often that's the option. If it's spreading so fast, you can't operate to remove enough tissue, so you have to take the leg. That's another one. This is on the top of the right foot this time, and again you can see the black kind of tissue. This is all dead. Hopefully this is fairly healthy here, but imagine trying to keep that clean in the hospital environment. Very difficult. Again, very rapid. Very rapid indeed. This is a slightly different one. This is gangrene due to C. difficile, Clostridium. C. perfringens. And again, massive damage to the kind of lower leg and the top of the foot. Now, I showed those slides, which aren't particularly nice, obviously, but these are situations where you can use a local approach if it's fast enough. Okay? And I haven't seen anyone use photodynamic approach to this kind of thing, even in an animal model. But surely this is the sort of thing that will kind of save, perhaps not save lives, but save the leg, save the foot, that kind of thing. So it's a very interesting situation. You can, obviously, even with a large amount of tissue like that, you can apply photosensitizer to that directly. You can illuminate, you can see it, you can see where the infection is. And, as I said at the beginning, very, very rapid kill in that case. I talked about mycobacteria and TB. There's all sorts of different diseases caused by this, including Hansen's disease, or leprosy, as it used to be called. These are very difficult to kill because they have a very waxy exterior, so slightly different from normal bacteria. But again, we can use even things like methylene blue and light will inactivate M. tuberculosis. I think there's a picture of TB coming up, so again, it's not a particularly nice one. So you can see the extent of the infection. This is a cross-section through the chest. See the kind of windpipe here. And all of these yellowy inclusions, these are all infected alveoli. And you can see how much TB is involved there. So, talked about this before, I'm not going to go through it all again. You can see the different drugs, conventional drugs, which are used for different sites, different specific sites, that's very important, in bacteria. So things like quinolones for DNA, they interact with the enzymes to do with stabilization of the DNA double helix, or the kind of traditional one, the beta-lactams, cephalosporins, penicillins, work at a specific site in the cell wall against the kind of cross-linking activity, which strengthens the cell wall. So if you can do that, then the cell will just pop rather than continue to grow. So these are very specific biomolecular sites, and this really is why, if there's a very small change in that particular site, the drug no longer works. So if you've got more than one site of action, or more than one mode of action, then you're more likely to get around any kind of resistance development. Just want to have a quick word about charge in terms of photosensitizers. There's plenty of evidence to show that if you have a cationic photosensitizer, excuse me, positively charged photosensitizer, then you will have activity against both types of bacteria, so gram-positive and gram-negative, both susceptible to positively charged photosensitizers. And it doesn't matter if it's a phthalocyanine or a porphyrin or methylene blue or whatever. As long as it's got a positive charge, then it's probably going to work against gram-positives and gram-negatives. Okay, so you can see activity here. This one is E. coli, This one is Staph aureus, which is a gram-positive. You can see activity for both cationic and anionic photosensitizers against the gram-positive, but against the gram-negative, in this particular experiment, we didn't see any activity at all against E. coli because 100 micromolar was the highest concentration that we had. So no actual activity there against gram-negative. And there are good reasons for that. But just again to say that it's not really to do with producing singlet oxygen, because you can see both the phthalocyanine and the porphyrin better producers of singlet oxygen than the phenothiazines here, and yet there's no activity against E. coli, and that's down to this problem with the charge on the photosensitizer. Okay. Now, it doesn't matter also whether this is Staph aureus or methicillin-resistant Staph aureus, MRSA, doesn't make any difference. Equally with E. coli, you could have something which is resistant to all of the kind of beta-lactams and the new carbapenems. Okay? Very common in healthcare these days. Doesn't make any difference whether it's that or susceptible, beta-lactam susceptible E. coli. Both of those are susceptible to photodynamic approach. Okay, so if we look at the target, and we can try and explain how this activity, this gram-positive, gram-negative difference in activity comes around. We have, again, this is a very simple diagram, gram-positive cell wall. Most of the gram-positive cell wall is peptidoglycan, but it has these extensions, these long kind of polymeric type acids called teichoic acids or lipoteichoic acids, and these contain negative groups. They're usually phosphate groups. So you can envisage that you would get a nice ionic interaction between a phosphate group and a cationic photosensitizer. Okay, with the gram-negatives, slightly more complex, well, quite a good deal more complex, but the kind of extensions from here are lipopolysaccharide, LPS, and it's kind of a lesser interaction with those which we see with the anionic porphyrins. We can actually dig down into the actual chemistry involved. Okay, so you can see, sorry, with the lipoteichoic acid and the wall teichoic acid, they're very similar kind of molecules, but you can see where the blue arrows are, sorry. Okay, and these are negative sites. Okay, so a lot more of these in gram-positives than in gram-negatives, and you will see a nice, strong interaction. And obviously, this is the basis of the gram classification, because the gram classification uses a cationic dye, crystal violet or gentian violet, and that is what will stick to gram-positives rather than the gram-negatives. It still will attach to gram-negatives, but it can be removed, which is the basis of the classification. Okay, and gram-negative cell wall, I've just included that for completion. The numbers of phosphate groups here are not anywhere near as big as with the gram-positives. Just for completeness, this is a schematic of Aspergillus, which is a fungus. And you can see, again, there's a membrane here, and you've got this kind of cell wall on top, a different structure to a bacterial cell, and so, of course, explains why conventional antibacterial agents don't work on Aspergillus, so we need an antifungal agent for that. And usually, as I said, they work on things to do with the membrane. So they obviously can get through the cell wall, but they have to act on this part in here. So what we're talking about in terms of cell damage, a very simple diagram here. Photosensitizer, excited, triplet state, and then we get reactivity. So we're looking at nucleic acid, or protein, or membrane, or cell wall. And it doesn't matter whether we do it from the inside, or we do it from the outside, or we do it from both, because the reactive oxygen species that we produce are so highly oxidative that they react with all sorts of different molecules within the cell and cause damage. Now, we'll look at the kind of chemistry involved, so we actually know what the molecules are and what's produced. If you think about DNA damage, this is what we normally would see. Okay, so guanine, this is guanine here, cytosine base pair, the kind of classic base pairing that we see in the double helix and all the rest of it. The double helix, of course, held together by the hydrogen bonds between the different base pairs. If we then just kind of take guanine out of there, if we react that with singlet oxygen, what we get is 8-hydroxyguanine. Okay, you can see the DR is just like the bond to the rest of the polymer chain there. This can then rearrange or react further with singlet oxygen, and you get these things which no longer look like a pyrimidine type base. Okay, so you can imagine that the hydrogen bonding, which is very important for specificity and reproduction and all the rest of it, those, once you make this, the hydrogen bonding is different. Okay, so you actually get hydrogen bonding between two pyrimidine, sorry, the one I've gone blank. You can get reaction with, bonding with adenine instead of the cytosine that should be there, so the DNA can alter its structure in that way. If you have further reaction, then these no longer will hydrogen bond at all, and that's the kind of basis of the DNA scission, so the strand break. Okay, so the chemistry of the kind of DNA interaction is very well understood. Okay, if we look at amino acids, there are other ones. I've just used those ones for demonstration purposes. So we've got tyrosine here, tryptophan, and histidine. Obviously, these are all aromatic amino acids. If you imagine them as part of a peptide, whether it's a structural peptide or an enzyme, and then reaction with singlet oxygen causes this. And you can see that these no longer kind of look like what they're supposed to look like. So again, if you think of the function of the peptide or the enzyme, then you destroy the function because the structure is no longer there. Okay, so the tertiary structure, quaternary structure, would be at least disturbed, if not destroyed, by that kind of process. Okay, so damage to DNA bases, damage to amino acids, peptides, is all part of it. Now, if we look at this schematic here, this is about the fungal sterile biosynthesis. Really, the basis is production of ergosterol, which is very important for things like membrane fluidity in fungal cells. But if we react this kind of biosynthetic pathway with singlet oxygen, we get these things. So none of these is what it's supposed to be in terms of the biosynthetic pathway. So there's so many kind of double bonds in these things. These are very receptive to reaction with singlet oxygen, and indeed with other types of reactive oxygen species. But these, again, the kind of potential for damage, for stopping the sterile biosynthesis is very high. So wherever we look in terms of biomolecules in the target cells, then we see damage with singlet oxygen. I concentrate on singlet oxygen, as I presume you've noticed, rather than the other reactive oxygen species, really because there is hardly, I've not seen any real resistance in terms of singlet oxygen. It's far more oxidative, far more reactive than the other reactive oxygen species that you see. So the idea is kind of sniper for the conventionals or grenade in terms of the photosensitizer. Okay? So with our conventional drugs, we're aiming at a very precise, very tight target area. So any deviation from that target area means we've got resistance. Okay, so that's if we've got very specific approach. If we have a photosensitizer, depending on where it is in the cell, of course, it will produce locally reactive oxygen species, and then they will go on and react with whatever they come up against. Okay, so using the chemistry that we just kind of saw. The other point that I made earlier is that it's very rapid kill. It's also non-specific kill. If you have something like a beta-lactam, penicillin or cephalosporin or carbapenem, these will only work if the cells are in the exponential phase. So in other words, if they're producing cell wall, trying to cross-link cell wall. Okay? If they're not doing that, then there's no reaction because the enzyme involved isn't around. Photosensitizers will kill or damage the cell whichever phase. It's completely non-specific. It doesn't rely on the presence or absence of an enzyme. And indeed, it's fairly well established now that enzymes are inactivated by singlet oxygen, even antioxidative enzymes. So things like catalases are inactivated by singlet oxygen because they're not, there's nothing there that can actually damage singlet oxygen. But an enzyme is just a protein, and we saw what happens with the kind of amino acids. Now, this is just to show you what you would see in the lab if you're kind of developing photosensitizers, as I have been since whenever it was, 92, or thereabouts. Both of these Petri dishes have been treated in exactly the same way, seeded with Staph aureus. The only difference is that this, after the seeding, was covered in foil, so it was in the dark. This was not. And both were illuminated with red light, same light dose, same time, and then kind of grown on. And as you can see, absolutely no cells at all concentrations that were used in that half, and plenty of cells growing in the kind of non-illuminated one. And this is just to show you that this is a drug called DO15, which I actually developed even earlier, probably about 1989. Initially, when I was working as in drug discovery for PDT, anti-cancer PDT drug development. But it's so good in terms of microbial cell kill that we try to use that as, again, a lead structure for new compounds for antimicrobial. So this is phenol. So a kind of an oily layer, and this is water. We put the DO15, this compound, in water. It gives you a nice kind of dark blue color. If you add octanol, then you can extract into the oily layer. So this, if you had methylene blue, you would only have a blue color in the aqueous layer and completely colorless in the oil layer. So it tells you something about the structure and the behavior in solutions. It's very important. Quite often we need to interact with membranes. So membranes require a little bit more in terms of oil solubility or lipid solubility. Okay, so again, this kind of helps with that. And it's very highly active against gram-negative bacteria, not surprisingly. Now, I managed to find this last week because I thought you might like to see actually what happens to cells. This is absolutely not my work. This is from a Chinese group published in Frontiers in Microbiology, 2016. The compound that they used is a porphyrin. And as you can see, porphyrin has 4 lysine chains on it. So it's at least tetracationic, 4 positive charges. And the work was based on a gram-positive, MRSA, gram-negative, Pseudomonas aeruginosa, and another standard gram-negative E. coli. So you've got the bacteria without any light or any photosensitizer, and then you've got the bacteria with photosensitizer but without light, and you can see even there that there is some change. So there's some interaction between the photosensitizer and the, presumably, if you look at this one, presumably with the outer membrane in the gram-negative. Okay, so it will cause a little bit of swelling, so there's probably some membrane damage there. And then when you illuminate, it looks like it blows the membrane apart, and you've got, I guess, bits of membrane here, but also parts of the bacterial cell will kind of come out. So that shows you the kind of cell kill, the damage, the physical damage, at least, to the bacterial cell. Okay, I think that's as far as I'm going to go today. Are we going to ask for questions at this stage? Yeah? Okay, that's fine. So, any questions?
Hello, thank you very much for your presentation. I would like to know, like, why is there less resistance to singlet oxygen? Is it because superoxide dismutase is not well expressed in bacteria?
No, it will inactivate superoxide dismutase. Superoxide is fine. So if you've got superoxide and you've got superoxide dismutase, that's okay. That will.
But what will be the difference then between human and bacteria? Because I know that some resistance to singlet oxygen in human cancer, they upregulate this antioxidant system of superoxide dismutase.
It may be that there's less expression of superoxide dismutase. I don't know. Okay. But there is no antioxidant resistance in terms of microbes. So whether it's SOD or catalase or peroxidase or whatever, okay. So it doesn't seem to work because it's presumably because of the high reactivity, and if you have a high concentration also of singlet oxygen, for example, then it will just react with whatever it reaches first, I guess. So it'll damage the amino acids. It may not even get to the active site where you would see deactivation. So the whole thing is chewed up. It is published work in terms of antioxidant enzymes. Okay.
Okay. Thank you.
Questions? Thank you so much, Professor Mark.
You're very welcome.